In terms of extraction of mycotoxin, in terms of method implementation, to be efficient and to prevent errors in the detection of mycotoxin, one should refer to the quality insurance principle. The analysis should then be performed by a degree of uncertainty. Certain sampling techniques, if carried out improperly, will affect the sampling and will further induce also dramatic errors. Training of personnel seems therefore mandatory to prevent those side effects. Then the pretreatment step of the sample should prevent inaccuracies through the use of proper instruments and proper tools. Finally, the analytical procedures must be performed by a sufficiently trained technician. It's important that tests use the appropriate and certified reference material together with a validated method of analysis to guarantee final result and good quality should be used. On one hand, rapid and sensitive detection is generally needed, using screening tests that are cost-effective and easy to use and that are available to anyone in the field. Generally, the drawback is the lack of selectivity, the cross-reactivity, fluctuation of the response according to maybe some variation of environmental conditions such as pH, temperature, matrix effect, and competition between mycotoxins. They further require confirmation with a more sophisticated method that is able to show the precise speciation and quantification of mycotoxins. In this respect, the analytical detection of mycotoxin can be approached from different angles, using different separation methods such as gas or liquid chromatography. New technologies are now proposing ultra-fast or ultra-pressure elution of the analyzed through a capillary-based or high-pressure system, diminishing the length of the HPLC analytical run. Then, a multitude of connective detectors can be chosen for the determination of mycotoxins. For example, we can choose between fluorometric or diode detectors that are the most popular types of detectors used in this field, and according to the referenced literature available in the official database. Some of the things to consider about these methods are accessibility of the instrument, there is a very high throughput for quantitative analysis, and low interaction with the matrix. The main limitation of this method is in the need to use specific chromatographic separation condition in relation to the analyte being investigated, provided the molecule fluoresces, otherwise it further requires derivatization of the sample. Other challenges can be recovery levels, the stability of the derivates, and overlapping of eluting compound. Thus, a multi-componential approach for rapid detection of multiple analytes suited for various matrices is required. The two-dimensional mass spectrometry, if unaided to separative chromatography, is then becoming the new method of reference for mycotoxin analysis. This approach allows the speciation through atomic mass of all the elements present in a sample and a clear mycotoxin assignment after specific fragmentation pattern experiment. While many components have the same intact mass value, adding another dimension through fragmentation enables to specifically fingerprint each sample that is analyzed. Quantification is enabled by specifically monitoring the multiple fragments over the chromatographic elution time. Analyses are ultimately increasing in sensitivity and precision. The addition of such a chromatographic step enables the time-dependent and elution of the species, further proposing multi-component detection of mycotoxins within a sample. Some of the limiting factors are suppression or enhancement of ionization, and this is generally due to the matrix and the huge diversity of mycotoxins that can be found, in addition to the lack of any calibrant. Recent work enabled the detection and dereplication of near 500 different fungal metabolites, albeit providing only qualitative results. Another interesting aspect of LC-MS approach is the possibility to work directly with raw extracts, usually obtained after solvent extraction of the sample and really basic filtering, which enable a significant speed-up of the analysis. Another major problem is that mycotoxin can be found as conjugated metabolites to one or several glycoside, for example, or sulfonide or glucuronide residues. Those different conjugates are masking the mycotoxin from being detected with even using an analytical procedure. The constant evolution of the analytical device and the extraction procedures will help mitigate these kinds of errors, and as well as better understand, predict, and spot the mycotoxins that are occurring in the field.